have a lot of interesting talks lined up in this day's sessions. First among them is on the infectious diseases. To chair this session, I invite Dr. Ashish Das. A brief introduction of Professor Ashish Das. Professor Ashish Das obtained his PhD from National Institute of Immunology, New Delhi, India. He has worked as a World Health Organization Fellow at the Malaria Branch Centers for Disease Control and Prevention. Professor Das has been associated with the Department of Biological Sciences at BITS Pilani, where thrust areas of this group is research into molecular biology and immunology of the human malarial parasites, especially using samples obtained from clinical cases. I warmly welcome Dr. Das to chair the session on infectious diseases. Welcome, sir.
Good morning, everybody. Welcome to the first session of the day. It's my privilege to invite Dr. Stella Shenek, who is a professor from the Universidad Ricardo Palma with a doctorate in microbiology from the Arizona State University, a postdoctorate at CDC in Atlanta, and from Harvard University School of Public. She has worked in the surveillance of parasites resistant to antimalarial treatments and in studies of therapeutic efficacy. The main objectives of Dr. Stella's line of research is the construction of a program that allows the development of new diagnostic tests and the implementation of surveillance platforms for the control of infectious and neglected diseases.
Thank you very much for the introduction. It is my pleasure to be here at this Genomics event for the first time in India. And I want to thank the organization committee for the invitation. And today I want to share with you some of the work that we've been doing in Peru on building capacities for genomics and tracking infectious diseases. So just to give you some background, this is my home country, Peru. I went back in 2019 after being trained in the US under a program that was sponsored by the World Bank and the Ministry of Education. And the idea was for me to help in the implementation and building an Institute of Tropical Diseases in the Amazon region, in this part, in the northeast part of Peru. Additionally,
much sooner than some of the alternatives, than doing individual tests. So this is something that we are excited about, and we think that there is an opportunity here. So there is an opportunity to enable more testing, and there is an opportunity to build new use cases like what we are doing with a small number of assays, right? 9 assays, but able to catch more than 200 targets. So these kind of novel use cases are also possible. The other audience I want to speak to are people in the audience who might be making novel assays, and I want to stress that this is...
So it is how to pick up, and they want to know that what was the sex of the this mummy, and they tweaked the protocol and used this NEV next Ultra 2 DNA library preparation kit, and result indicates that they were able to identify the gender of this 4,000-year-old mummy. Actually, this is highly degraded DNA. If you see, this kit is already working with the degraded samples, FFP samples, but still there are labs which are working with FFP, and you know that FFP is already degraded. But there is not only the degradation, but there are other things which are happening to FFP samples during the fixation. That, for example, deamination, nicks and gaps.
genomes, coding sequences and gene feature files deposited for all of them. Like for human, mouse, rat, all of those things. And then for de novo analysis, some of the organisms, say like you have earthworms or certain rare plant species where you don't have the genomes which are deposited. So if we don't have the genomes, we go for the de novo assembly, where we would assemble the reads and then use them for analysis. For reference base, we directly take the genome and align the data to that reference genome and go for downstream analysis. So I'll explain them to you step by step. Yeah. So now starting with the first step. First step is the raw data. Raw data is basically the data what we receive after sequencing from the NGS lab. And first thing what we have to do is we have to check the quality of that data, if that data is of good quality or not. So to check that, the tool what we use is FastQC. In my coming slides there's a FastQC image, so I'll explain in detail how do we analyze the FastQC. Apart from that, we also have TrimGalore. TrimGalore is basically used for two main things. First would be removing the adapter sequences. So when we are doing Illumina sequencing in the lab, there are adapter sequences which are there. We don't need them for analysis. So we remove the adapter sequences. Second thing is we remove the low quality bases. So I'll just explain that. So for transcriptome, the chemistry of the data is 150 cross 2. And what basically you mean by 150 cross 2 is a paired end data. So your sequence would be of 150 bases and cross 2 means paired end data. So if you see this particular diagram, you can see read 1 which is from 5' to 3', and then in the other direction, same the complementary you have 5' to 3'. So this is known as the paired end data. And the data what we receive is in the FastQ format. So I think yesterday also we saw what exactly a FastQ file format is. There are
in the number of mutations that there are in the tumors of these patients. Again, we've done, like I said, that we've done whole genome and whole exome sequencing, and we have identified mutations that are drivers that drive the cell to becoming a malignant tumor. There's a lot of mutations happening at the background, and we need to weed out those mutations in order to catch hold of the drivers. And what we have done is to be able to catch hold of the drivers. Some of these genes may actually be familiar to you. PP53 caspase 8 FAT1 may or may not be familiar to you. So there are some genes that are known from earlier from other kinds of cancers, but there are also some genes that we found to be significantly mutated in and recurrently mutated in a large number of patients, and those genes are not so popular genes. All of these genes, some of these genes can behave both as tumor suppressors and as oncogenes, but all of these genes behave as tumor suppressors in the present case. I don't have time to show you the data. If you look at those genes, if you look at the driver mutations in these genes, and we ask ourselves, do all patients look similar? The answer is no. You can actually find three subgroups of patients. In the first subgroup of patients, inspiring meeting
and I thoroughly enjoyed looking through the program and really getting a sense of the depth and breadth of topics that you covered. And in many ways, I think it's appropriate that I'm sort of giving the last talk because I'm going to give a very broad story about the history of genomics, past, present, and future. And I think I'm going to leave you at the end with a very much of a feeling where we think things are moving in a very exciting way. Now, I'm going to give you my version of the story of human genomics, and part of it is because I've lived it, basically from the beginning of the field, but also because, particularly the last 13 and a half years, I now lead the National Human Genome Research Institute, which is part of the U.S.'s National Institutes of Health. And we're the major funder of biomedical research in the United States, and actually my institute, one of the 27 institutes at NIH, focused millions of times now, and we're learning more and more and more about the genomic signatures of changes and aberrations in tumor cells that really give us clues about the mechanisms of cancer.
From IIT Delhi. And at the first place we have Pratibha, CSR IGIB. Congratulations to all the winners for the NDSA test. We also had a bio quiz during the time the conference was being planned and organized, and we have winners for that as well. The third, Astha Rai, St. Joseph College, Bangalore. Congratulations on the winners. Coming to the third part, we had some excellent posters presented during the conference, the 2 days, and they were evaluated by Professor Pan microbiome in comparison with rural, non-rural, and urban population. The second place goes to Priyanka Kumari, Institute based classification of E. coli sequence types, implication and new findings. Congratulations. Exciting evening.
like. Yeah, I invite Dr. Raja to just say a few words now.
Thank you, everybody. Whatever has to happen has happened. And the other people, I will start with my friend Dr. Pandian, Rakesh Mishra, Professor Doran Lancer. Here is my senior, Professor Kannan, and so many other people whom we call as old monks. Being here, Dr. Sharon, Dr. Mithu, Vaniji, I think many people, many people who came here, Manigaru, all of them with their moral support, they are there, and Dr. Ashish Das is there. So, thank you all of you for So finally, all good things has to end, and it's ending now. But I think this end is going to be a great beginning for everybody. The students who all decided they're going to delete or reset the computer, reformat it and put Linux and start doing NGS. So many of you here.
Thank you, Raja. I know it's been a very long information overload, and of course you've had a lot of opportunities to interact with speakers. We sent out invitations to many people across the globe, some of whom could not join us because of visa issues, but we still had many people from Bangalore, from across many countries, and across India. We conceived of this, I think, about 3 months ago. This was, 3 years of COVID did not allow us to continue doing the sessions which we started in 2014 and 2019. Essentially, this also marks 25 years of our existence as Genotypic. Human genome, quite a remarkable year this is, and we thought this was a befitting time to do the conference, and the concept and idea evolved over the last 3 months, but I think without the support of many people.
Believe that actually 3 months back, that this will take such a fantastic shape. Thank you, Sudha, thank you, Raja, thank you,